In this video, we will discuss about role of glycans in the cancer hallmarks. So, basically, we will cover most of the points that we have covered in details in one of our previous videos, or in many of our previous videos. So, we will start with this sustained proliferative signaling. So, in this case, what will happen is the aberrant glycosylation in the tumors leads to the upregulations of the upregulations of the genes that are responsible for proliferation. And one of the example is shown here, that is C. myc uh, get glycnac is glycosylated, and Fox M1 and cyclin D1 get glycnac glycosylated, and its combined complex structure leads to the proliferation upregulation of the genes that are involved in proliferations. Next thing is this resistance to cell death. So in this case, that we have covered this beta-tin-K AKT pathway many times in our previous videos, in which the phosphorylation or activation of this alpha 3 beta 1 integrin leads to this P3K AKT pathway activation, and that will trigger the upregulation of the genes that are involved in the survival of the cells. And similarly, this trial and fast path and kappa beta phosphorylations also works towards upregulations of the survival genes. The next thing is this deregulated cellular energetics. So in this case, the glucose consumption pathway and the lactate consumption pathway, or in general terms we can say that energy consumption within the cells is reprogrammed in a way that it will promote the uncontrolled cell growth, or it will support the uncontrolled cell growth even in these hypoxia conditions. So in the case of hypoxia conditions, this HIF1 alpha factors get upregulated, and it will trigger HIF1 alpha targets genes. Next one is this evasion of the growth suppressor. So we know that one of the characteristics of the tumor cells is the uncontrolled growth rate. So this uncontrolled growth rate is happened because of this because this growth controlling capacity of this growth suppressor got disrupted in the case of tumor cells. For example, this P53 has a functions to decide whether the cell should go for apoptosis, or it has a capability to repair its cells. So this P53 apparent glycosylation of the P53 leads to this dysfunctional of this P53 in a way that it will upregulates the genes that are involved in cell cycle progression. Thereby the cell cycles of this tumor cells will be at a higher rate compared to the normal cells. Next thing is this genome instability and mutations. So in this case, since we know that most of the tumor cells are mutated, so that mutations might even lead to this DNA damage, and this DNA damage should trigger cell death through these P53 functions. However, P53 is not functioning properly. At the same time, there are other aberrant glycosylation of this ATM that should have leads to these proteasomal degradations. However, because of the mutation or oxidative microenvironment, this aberrant glycosylation of ATM prevents its proteasomal degradation, and thereby this this dysregulations of this, or the incapability of the tumor cells to uh, degrade these ATM proteins will cause genomic instability and mutations, or we can say that uh, unbalance of the ATM glycosylated proteins within the cells cause genomic instability, and it will further promote the mutations within the tumor cells that will cause the DNA damage. Next thing is the angiogenesis. Angiogenesis is important for the tumor cells because the tumor cells needs a new source of new source or new source for the food, so that they can maintain their uncontrolled growth rate. These things can be maintained by this glycosylation of these VGFR2 receptors. When it's glycosylated VGFR2 receptors interacts with this galectin 1, it will trigger this beta T negative pathway, and that will not only upregulate survival genes, but it will also upregulates the genes that are involved in angiogenesis. Thereby, angiogenesis, it will trigger this angiogenesis, such as will trigger more blood vessels, uh, more blood vessels to the more vesicles to the blood vessels, so that the blood supply can be established, and tumor cells will be able to fulfill their requirement for the uncontrolled growth rate and their cell cycles. Next thing is this evasion and metastasis. So in this case, whenever because of the changes in the glycosylation profile, cells will acquire these EMT characteristics, epithelial to mesenchymal characteristics, and when it reaches to the secondary organ, it will again change from EMT to MET, that is mesenchymal to epithelial characteristics. So in this case, the figure shown is about when the cells, the tumor cells reach at the secondary organ sites, that this epithelial markers, that is E-cadherin glycosylated, will help in the binding to the EP selectins near the secondary organ, and thereby helps in the intervasculations of these tumor cells at the secondary organ site, and that will cause the metastasis. The next factor is tumor promoting inflammation. So since because of the aberrant glycosylation that has continuously occurred within the tumor cells, it will lead to this glycosylation structures or patterns, uh, or carbohydrate structures or patterns that are uh, that have non-human epitopes, and thereby it will continuously triggering these inflammations, and that will cause the continuous immune response at the site of tumors, at the site where tumor cells are more accumulated. The last thing that is that has a role of glycan in the cancer hallmarks is this immune escape, and this thing that we have discussed in our previous video, recently published previous videos, in which case the glycan structures such as Siglec ligands can interact with the Siglec receptors on the immune cells such as NK cells, and deactivate the NK cells, and simultaneously these dendritic cells will also be get deactivated through this checkpoint inhibitor, that are available on the dendritic cells, so by which the immune response against the tumor cells will get suppressed. So these are just a general brief overview how the glycans play a role in the cancer hallmark. If you need to understand in detail about the role of glycans in the cancer hallmark, you can go through the reference that has been provided in the description. That's all for this video. Thank you for your time.